select our B cells data here. So we think these are probably a lot of, if not most of the B cells in this data set. And then we go to the Analyze tab here and run the clustering algorithm and maybe try and define, I don't know, 5 different clusters of B cells based on our principal components that we already calculated. So this is just one way of doing unbiased clustering. And there's lots of different plugins available to do this as well within SeqDeep. But if I run this, I'm hoping that we get 5 different clusters back. Actually, I'm not seeing 5 different clusters. I apologize, that didn't work very well.
Now, are you gating on clusters of VDJs?
No, not yet.
Oh, okay.
I'm sorry. I just want to arrive at something for us to do a comparison.
Okay, right. Okay, good idea. Okay.
So let's try phenograph. So this is another clustering mechanism. And the authors of this algorithm claim that they can do a better job predicting phenotypes.
Oh, I remember. And you said at one time or another that doing clustering, graphing or whatever on principal components is a good thing to do. I remember you saying that. So you're not just looking at cells, you're doing principal components. Why is that better again? I mean, I can imagine, because the principal component includes, contributions from reduced in principal component space, a little bit of contribution from a lot of genes, and then they distinguish one population from the next. But why is it that just t-SNE plot, don't they, the t-distributed, why doesn't that sort of do the same thing?
So, t-SNE comes after doing principal component analysis typically.
Right.
The reason for doing the principal component analysis is to compress or condense the data matrix into something that's more amenable for these machine learning algorithms to work on. If we just fed those machine learning algorithms, like, for example, t-SNE, the raw data, we would get back a very strange looking set of clusters, and it probably wouldn't be, useful information. So we first do principal component analysis and get everything down into a more condensed data matrix, as opposed to the sparse matrix from sequencing.
I'm just thinking the loop, that program, the other program that 10x sends with that they sort of they made to do this.
Right.
They do t-SNE plot, I think, straight away.
Right.
And I don't know what and I look at that and there's a lot of clusters. Or you're saying those, but they don't do principal component first. You know what I mean? So they do principal component first. They just do it without any user input, so you can't change the. Oh, okay, all right, that makes sense. Because I figured if there's a better way to do it, they do it too. But so everybody does this. They do principal components, and then you do t-SNE on the principal components.
Yeah, it's called PCA guided t-SNE.
I see. Now the thing is you can select the number of principal components.
I can select the number of principal components. I can determine how many genes go into that.
Right.
I can even select subsets of cells, rerun PCA, and then do t-SNE again to get clustering within clusters.
Right.
So you can do a much deeper analysis within SeqDeep than what Loop provides. Yeah. At least that's what I've found.
Now,
but I have played around a bit with Loop.
The thing is, when you select the number of principal components, you select 15 or 20. What if you select 10? Does it matter how many you select?
It does to some extent, although we have found, after about 12 principal components, you're not adding a lot of, not changing the t-SNE clustering very much.
Okay. It seems like why I don't like the computer to work too hard. Yeah, right, I've got this, I've done this, and then they really get all over the place like this. I noticed that. And I'm sort of surprised that these B cells have so many clusters. But this is exactly where I was, and now I want to know what each cluster is, and what the immunoglobulin expression is in this cluster versus that cluster. You're exactly at the right place here.
Yeah, so at this point, we can begin to dig into these clusters and ask those kinds of questions, like what are these clusters?
Right.
In this case, these are clusters within what we think are B cells.
Right.
So these are probably B cells, doing different things. Could be plasma, memory, naive, different kinds of plasma cells. We have to explore further to get into that. But just this gets us to the point where we can start to ask questions about the EDJ. I might just save this analysis because I'm kind of interested in it at this point.
Mm-hmm. Can you, save this and of course, if you send it to us, is there a way just to? I see it over here. Every cluster is color coded. I don't know. Yep. I can't quite see it in detail because the resolution is not so good. But is there a way now to save each cluster over? I guess there is somewhere over here. I guess it would be in a, I don't know, cell window or gene window. I'm not sure which window it would go into. Oh, that looks, pick big dots. How can you save each of those clusters so we can look at each one?
Yeah, so each cluster is defined here. I named them phenograph clusters 1 through 11. So there's 11 different clusters there. And there's a lot of different things we can do with them. The next thing I was hoping to do was kind of identify the different clonotypes that exist within some of those clusters.
Yeah.
So that gets us into EDJ. Is that what you mean?
Yes, that'd be wonderful.
Okay.
So I was thinking of even what's the phenotype of the B cell or what the gene collection is in each cluster. But
right, we could do differential expression analysis on each cluster and get back a upregulated gene set that kind of defines each of those clusters.
Yeah.
But that's we kind of went over that a little bit, so you saw how we can create volcano plots to do that.
I see. Right. Okay. So we'll try that. And now we're going to look at the VDJ. That'd be good.
Yeah, let's see how this works. Maybe. Seems like it's taking a little while to open. It's not a good sign.
Now this you can do. We can do this up here. Because I kind of did this. You get this. These are down here in a cell window.
Yeah, this is the scary part.
Try and clean this up a little bit and see if I can get this to run. One more time here.
Are you going to do all the clusters? I mean, it'd be great if you can do them all at once. To look at the...
Exactly. I was hoping to look at how each of the clusters' different clonotypes look. Though it doesn't seem to want to run at the moment. Try one more time.
Wow, too hard. Too much power.
Maybe just try one cluster?
Yeah, 2. Compare 2.
Oh,
And it shouldn't matter. We've done this with really large data sets, and it should be able to. All right. Well, what I'm going to do is restart this. Let's kill it here. Quick restart.
Are you still there?